Welcome our next esteemed speaker, Professor A. Sujit, for the next lecture session. So, Dr. A. Sujit is a professor at the Department of Chemistry, NIT Calicut. He's a renowned expert in polymer and material chemistry with an impressive academic and research journey. He earned his PhD in polymer chemistry from NIT Calicut under the guidance of Dr. G. Unnikrishnan, followed by a prestigious postdoctoral fellowship at the Advanced Industrial Scientific Technology, Japan. His research excellence is reflected in 92 high impact publications, about 3,000 citations and an H index of 24. Beyond his research, Dr. A. Sujit is a dedicated academic leader serving as the secretary of the Technology Business Incubator at NIT Calicut since 2022, fostering innovation and internship. He is serving as a government body member of the Institute of Human Resources Development, IHRD, Government of India since 2017. He is a faculty in charge of the ISTE NIT Calicut chapter, empowering students and educators. His excellence in teaching and research has earned him numerous awards, including the Best Young Teacher Award from NIT Calicut in 2010, the Most Citation Award by Springer for his outstanding review paper in analytical and bioanalytical chemistry. So, with this introduction, let me welcome Professor A. Sujit for the lecture on spectroscopic characterization of biocomposites.
Thank you. Thank you for your nice introduction. And I'm very happy to stand in front of you in this prestigious platform of GAN, especially the discussion on biocomposites. So today, within this limited time, I want to discuss about the spectroscopic analysis of polymeric systems. So in this speech, especially I will be discussing on Raman spectroscopy. It's a very powerful tool for characterizing different type of polymeric systems. Okay, so let me start from the very beginning of the concept of spectroscopy. And can you imagine a situation where there is no light? What happens? How our life will go on without light? You see, we cannot see the beautiful chameleon without light, and also we cannot enjoy the beautiful wings of the butterfly. So all these things are possible because of light. That means the interaction of light with matter. And you can see there are so many applications, especially the reindeer. So how can we save the reindeer? Because this problem is due to the hitting of reindeer when it travel crossing the roads. So in order to avoid this in the night time, they have used a fluorescent material to apply. They are applying the fluorescent material on the reindeer, and then during the night time, the vehicle or the drivers can detect the reindeer and avoiding this one. So all these things are due to the interaction of light with matter. Okay, so I told you that the spectroscopy is a very wonderful technique for the characterization of molecules. Actually, it deals with the interaction of radiation with matter or the molecule. So when light falls on molecule, you can see that there are two effects. One is the absorption of light by molecule, and it will go to the higher state, and you know that it cannot stand for a long time at the higher state, it will come back. So at that time there will be emission occurring. So most of the spectroscopic techniques are absorption spectroscopic techniques, and the very important technique in this category is infrared spectroscopy. You already studied about this spectroscopic technique. And all these things are possible due to the light I told you, we have a wonderful range of the electromagnetic waves, and we are here actually in the visible region. Actually, we are going to discuss about a method that is making use of the visible light. There are other lot of techniques you can see: X-rays, gamma rays, UV, IR, microwave, radio. All these things are, all these type of lights are using, radiation is using for spectroscopic techniques. But we are very interested in this region, that is the visible region. You see, this is the picture, the schematic representation of a simple spectrophotometer. It consists of a light source and a wavelength selector, and your sample you will keep here, and your detector, and finally the recorder. So, this is a very general structure or schematic representation of a spectrometer. So, I am just want to share one of the problems faced by a very important industry, polymerics. Okay. So, polymerics is a polymer product company. So, they are mainly making spinal implants. So, these spinal implants are made by a polymer called PEEK, P-E-E-K, polyether ketone. So, they face the problem after the sterilization. So, normally before the implant, this is a spinal implant, and they will do the ethylene oxide sterilization. You know that ethylene oxide is a gas, and this type of sterilization usually doing in for the medical devices, especially for the implants. And after the implant, they are actually making this surgery, and there were lot of post surgery problems, and after the examination they found that normally after the sterilization the cracks are developing on the PEEK, this material. Okay. So, they have checked many they have checked it for analysis. That means FTIR, they checked FTIR of this compound, but they could not see any chemical change after the sterilization. And they have checked the SEM, scanning electron microscopy, and it actually revealed cracks. Okay, we can see, but we don't know why this crack is happening. Okay. And also they have checked for DSC, scanning electron, differential scanning calorimetry, and they have reported some normal crystallinity. Normally these crack type problems will be occurring due to the crystallinity related issue, but that was also not the issue. So, they have checked this material under a confocal Raman micro spectrometer. I will explain what is the technology. Okay, so they have checked their sample under a confocal Raman micro spectrometer. After that, they got so many results, they got some critical findings. Okay, the findings was, one finding is a new peak at 1610 centimeter inverse, and it assigned to the aromatic ring deformation. The peak structure, you see the polyether ketone, it has aromatic rings and also one carbonyl peak, carbonyl functional group, and also ether functional group. Okay, so one finding was a new peak at 1610 centimeter inverse due to aromatic ring deformation. Then the second one is the carbonyl peak has a shift from this 1645 to 1630 due to a hydrolytic degradation. So, actually this hydrolytic degradation is the main problem here. After ethylene oxide sterilization, some moisture is actually trapping inside the peak system, and finally a localized hydrolysis is taking place, and it actually affects the amorphous regions of the peak, and the crack is developing. Okay, so this type of analysis, this type of result they got from the Raman spectrometer. You see, there are a lot of advantage with this type of technique compared to FTIR. FTIR. So FTIR, that means the infrared spectrometer, normally giving a bulk. Suppose you are analyzing a bulk sample, it simply gives a spectra, a combination of all these things they will give. That means you cannot make it specific, you cannot make it localized measurements by using infrared spectra. But Raman spectra, you can see that it's a non-destructive technique. I think that the problem is that the advantage is that you need not destruct the sample. Okay, so from the sample itself you can measure the spectra, and normally it works in all environment. So I am going to tell you a wonderful technique for the polymeric system characterization, that is nothing but Raman spectroscopy. Okay, so you see this incident is in September 1921. The SS Narkunda ship is traveling from Britain to India through the Mediterranean Sea. It was a sunny morning. You see the Mediterranean Sea is beautiful, blue color, and also the sky is also blue color, very attractive blue color. അപാര സുന്ദര നീലാകാശം അനന്തതേ നിൻ മഹാസമുദ്രം അപാര സുന്ദര നീലാകാശം. A very beautiful sky you see. It's actually look similar to the infinite ocean. Okay, so the sky and the blue has a lot of similarities. The very important similarity is the blue color. Okay, so this one of the scientists, physicist, he was a physicist. So he is actually came out from the ship, and he moved towards the deck of the ship with a glass of water in a sunny morning. Okay, so the scientist was, you know the scientist, okay. So this scientist is actually Sir C.V. Raman. Sir C.V. Raman. So he is actually looking into the blue sky and also the blue sea. Blue sea. And you know that what is the reason for the blue color of the sky? We know that, okay, because the well-known scientist Rayleigh, physicist Rayleigh, explored the blue color of the sky by explaining his famous Rayleigh scattering. Rayleigh scattering, that means sunlight is when pass through the dust particles, it will undergo scattering, and that scattering normally for us it look like blue. So why it is blue? There are a lot of colors because sunlight is actually undergo scattering. Sunlight consists of 7 colors, so all the colors will equally scatter, but we can see blue color. Why? Because the blue color is, you know, the wavelength is very low compared to red color, red region. That means if the wavelength is very short, means it can interact with a lot of dust particles. That means the probability will be very high due to the shorter wavelength. But our sky, normally you know that violet, indigo, blue. Violet is very short wavelength compared to blue, but we cannot see the violet sky. Why? that is actually our problem, because we can only see the combination of violet, indigo and blue, and it will be appearing as light blue for us. But that may not be it may not be same for the other type of insects or some other creatures. Sometimes they can see the violet sky. Okay, anyway, this the blue color of the sky is actually explained by the scientist called Rayleigh. Okay, Rayleigh. But that was not the problem here. The problem is with the blue color of the sea. Blue color of the sea. And Rayleigh has also an explanation for the blue color of the sea. It is nothing but the reflection of the blue sky. Blue sky. But the problem the C.V. Raman faced is, so he is actually standing on the deck with a glass of with water in the glass. Okay, he looked into the water and he could not see any blue color because the sky is there, and he is carrying the water like this, but the water is colorless. Water is not blue. If it is the reflection of the sky, this water should become blue. It's not blue. Okay, that's one of the problems. Okay, so he realized that the blue color of the sea is not due to the reflection of the sky. He analyzed water and also he analyzed a lot of other solvents, and he found that the same type of scattering is happening in the sea also. That means the sunlight is interacting with the water molecules, and it will also cause the scattering, and it will be appearing as blue. Then there is another problem. Okay, if it is true, why water in your glass is again colorless? The same sunlight is actually falling here, and molecules are there. It will be undergo scattering, but there is no blue color. So why there is no blue color again? See, water in our well is not blue color. Water in our river is not blue color. Water in the sea only blue color. Why? See, the problem is the number of molecules. You cannot compare the number of molecules in glass and the number of molecules in the sea. Okay, so when the number of molecules increases, the scattering effect will be also increasing, and it will give a wonderful blue color. Okay, so after that, there was actually a particular type of collision is hidden in the universe, that is known as the inelastic collision of light and particle. Okay, so the Rayleigh explained the blue color by the elastic collision between light and particle, or the light and molecule. Okay, what do you mean by elastic collision? Elastic collision means the energy before collision is equal to energy after collision. That is known as the elastic collision. But Raman thought of the other type of collision that may be possible. That is the inelastic collision. That is, energy before collision is not equal to energy after collision. That is also possible. But we cannot feel the inelastic collision because the major type of scattering is contributed by the elastic collision. That means elastic collision between light and particles. That is why Rayleigh scattering is very prominent, and normally you cannot see the Raman scattering. It is actually hidden in the Rayleigh scattering. But you see, Raman used a very old spectrometer like this, and he used a particular liquid for the analysis, and he passed the light violet, using a violet light, he used the violet light, and he could observe that there are two types of lines are coming. One is the Rayleigh line and Raman line. But Raman line we cannot see by our naked eye. But if you are using a filter, a notch filter or a optical filter, you can see that light. That is the Raman radiation. Raman radiation. Okay. So you can see the Raman radiation here. The light is passing through the liquid, and suppose you are applying a filter, you can see the Raman light. Raman light. It is just like you are beating a drum. Okay. So suppose the drum is there, and you take the baton. Suppose you are beating the drum first time. What happens is, actually I am transferring the energy from the baton to the drum. It's just like molecule radiation is, photon is colliding with the molecule inelastically. That means energy is transferring from photon to the molecule. But the second hit I am doing with a baton like this. During the second hit, what happens? You see, after the first hit, the molecule is, or the platform is already in a vibrated state. It's in an excited state. The second hit means one effect is, normally it will transfer energy from drum to the baton. Okay, so there are two types of inelastic collisions. So you can see that this is the incident light, and this is the scattered light. These are the energy levels. You can see that incident light will come back as the scattered light, but it will come to the this energy level, because the energy is transferred from photon to the molecule. So this much energy is transferred. Okay, but the second type of inelastic collision, you see, the molecule is already in a vibrated state, and it will take the incident radiation, and scattering will be more. So this two types of scattering will result in two types of lines in Raman. One is the Stokes scattering, and another one is the anti-Stokes scattering. But you can see one more line here. It's a very high intense compared to Raman. That is the Rayleigh line. That is due to the elastic collision between the light and particle. So one of the important problem we are facing is Raman scattering is a very weak effect. Okay, very weak effect. And also this part, that means the Stokes scattering, is mainly we can use for the analysis, chemical analysis. So you see, we are using this region, that is the Stokes radiation, for our analysis. Okay, so anti-Stokes scattering is also using, but it has limited applications. Okay, so this is the complete picture. That means the Rayleigh is very prominent. Normally the Raman lines are hidden, but if you are using a filter, you can see these two lines, Stokes and anti-Stokes. Okay, so we will go to our, gradually we are going to our problem. Infrared spectroscopy is a type of absorption spectroscopic technique. That means it depends upon the light absorbed. It is working based on the absorption of light. But Raman spectroscopy is very different from infrared spectroscopy. Actually, here we are considering the scattering of the light. The principle of Raman spectroscopy is the scattering of light. But we are enough lucky, because due to the scattering, at the same time the molecule will vibrate and also molecule will rotate. That is why we have vibrational Raman spectroscopy and rotational Raman spectroscopy. So we will make use of vibrational Raman spectroscopy. It's not a type of absorption spectroscopy. It's a very different type of spectroscopic technique. If you are just compare Raman and infrared, normally you see that same type of peaks you can see. Okay, so vibration If you are an expert in infrared spectroscopy, spectral analysis, you will be an expert in Raman analysis also, because the same frequency region you can see here, it will show the peak, but the intensity of the peak will be different, and also sometimes some new peaks you can see in Raman. Why it is? Because of the peculiarity of Raman spectra. Because what is the important criteria for infrared spectroscopy? One of the important criteria, that is the dipole moment change during vibration. That means dipoles or dipole moment change during vibration, that type of molecules will only show infrared. But Raman has no limitations like that, because Raman spectroscopy is not depend upon the permanent dipole moment. It depend upon the polarizability of the molecule. That means non-polar molecules also will show Raman spectra. So that is a very important principle that can be applicable to polymers, especially for inert systems. Okay, so how this type of polarization is happening? Suppose a molecule is there, you take hydrogen molecule, hydrogen has no dipole. Okay, H, there's no dipole. Okay, hydrogen is in an electric field. When you are using visible light, you see there will be one interaction taking place. That means the positive pole of the electric field is interacting with the negatively charged electrons, and the negatively charged electric field is interacting with the positively charged nucleus. That will produce an induced dipole moment, and this induced dipole moment is equal to alpha into E. Alpha means, alpha is the polarizability of the molecule, and E is the electric field. You see, Raman spectroscopy is mainly depend upon the polarizability change in a molecule. So polarizability is a very important property here, and polarizability is also directional dependent. That is also one of the advantages of Raman spectroscopy. That means directional changes or conformational changes and deformations, all these things will be showing by Raman spectroscopy, because polarizability change is directional. That means the nature is known as the anisotropic nature. Okay, so Raman spectroscopy can be used for the characterization of a large number of compounds, and especially we will be focusing on the carbonaceous materials and also polymers and also biopolymers. Okay, so this is the experimental setup of Raman spectrometer, and I will be first discussing about this thing. This is, you can see Raman spectrometer is here in this material science and technology. You can see a Raman spectrometer. A Raman spectrometer is normally connected with a microscope. That is why it is known as micro Raman spectrometer. And you can see the objective and eyepiece here. And this is actually the sample stage. And you can see a red light. No, this is the laser. So source of radiation is laser here, laser light. So laser light you can use, and this is your sample plate, and this may be the detector part, and finally the spectra will be displaying here. Okay, so when you go to the experimental side, you can see that it's somewhat complicated, but I'm just highlighting some things. One is the sample stage. You see, this one is your sample, and this is your sample stage. Sample stage is very simple here. That is the one of the peculiarities of Raman. So IR spectroscopy you have taken, no? FTIR. So there is the sample chamber is under vacuum condition, and the preparation of sample it needs some work. Okay, somewhat not so simple. But see, Raman spectrometer, this sample preparation is very easy. You can just take a glass plate, a neat glass slide, and put your sample, and you can cover it with a cover glass. That's all. Our sampling is over. And that you can take it in any environment. Okay, suppose your sample consists of water. Can you take an FTIR, perfect FTIR with moisture? You cannot take because moisture means H2O. H2O will give 3 prominent peaks in IR. That will be interfering with your sample spectra. So it will become more complicated. But here the important advantage is water is a non-Raman scatterer. That means it will scatter, but a little bit. So you will not get a prominent peaks for water in Raman. Maybe some peaks may be showing, but it is not so prominent. So in any environment, you can take the sample, okay, in water or moisture type. That is a great advantage here because you can analyze biopolymers. Suppose the biopolymers, dried biopolymer means it will lost its conformation. For a very good example is proteins. Protein will lost its conformation, and a lot of structural changes will be happening during drying. So drying is not needed here. So this is a sample stage, and you can under a microscope you can do all these things, and there will be 2 detector. One is for the image, and other one is for the spectra. Okay, so 2 types of camera detectors you can see. Okay, so why I am showing this one is in all microscopic techniques you can see this type of crosswires. So what is the need of this crosswire? Focusing. Suppose you have a lot of samples like this or some particles like this. You want to focus on this sample. Means you can just keep the crosswire here, and you can in Raman spectrometer you 2 advantages there. One is you can take the picture, and another advantage is you can record the spectrum. So the visual effect is more in Raman spectrometer. Okay, we will go to the technique first. Okay, suppose for a polyethylene. So there is no reactive group and there is no functional group in polyethylene. Simply it consists of 2 types of bonds. One is CC and other one is CH. Out of this, CH is polar, and also there will be vibration. The dipole moment change during vibration. So CH can be detected by the CH is normally can be detected by FTIR, infrared, but this C double bond C cannot be detected by IR. Okay, so you can see this may not be much visible. You can see this one. This one is the peak for polyethylene under the range of 3000 to 2500 range. Okay, maybe this portion is highlighted here. You can see prominently 2 peaks only. That is actually coming from the CH vibrations. But CC vibration you can see additionally in Raman spectrometer. So most of the polymers consist of CC bonds. The conformational changes, the deformation, all these things you can find out by Raman spectroscopic techniques. So that is one of the advantages of Raman spectroscopic technique. One more advantage is there. Normally for use for the biocomposites and for making different type of composites, we may be using carbonaceous materials, carbon based materials diamond, graphene, fullerenes, the nanotubes, all these things we will be using. You can easily characterize these type of carbonaceous materials by using Raman spectrometer. You cannot characterize these type of techniques by compounds by infrared spectrometer, but because the problem, what is the problem here? Only CC bond. There is no polar bonds. Polar bonds. The only technique which will tell you about the conformation is Raman spectrometer, Raman spectroscopy. Okay. So, for example, diamond. So, you know that a diamond only consists of CC bonds. CC bonds. So, it will form. It will show a strong peak, a CC peak somewhere around 1500, and this single peak is due to the uniform CC bonds in the tetrahedral structure of diamond. Okay, but it is different for the other silicon and germanium. All these things you see in the extreme side, you can see some peaks. Okay, so polarity is not a factor here. Okay, so this is the comparison between the diamond and graphite. So the graphite normally showing a peak called G band around 1582 centimeter inverse. So these are all the signatures of these type of carbonaceous materials. Okay. Okay. So one more important thing you see, the buckyballs, C60 buckyballs, it also consists of CC bonds. Okay. So due to the deformation, if some deformation is happening, so C60 to C70 means some deformation is happening. C60 is somewhat perfect, but when you are going to C70, it is somewhat deformation is happening. This deformation can be detected by Raman spectrometer, because this peak shift, the peak shift to the lower, higher frequency region shows the deformation. That means a lot of signs. This technique is very sensitive to this type of deformation and all. So that is the advantage of one of the advantages of Raman spectroscopy. And the graphene and graphite you can differentiate. You know that graphene is the single layer of graphite. So if you are going for other techniques, infrared spectroscopimeter, the same normally it will not detect because of the CC bond only. But if it is possible, means there is also one more problem, because the layer and all the bulk material it will consider as similar. But Raman spectroscopy is very sensitive, and there will be intensity change and small peak shifts for the graphene and graphite. Okay. And we will be, for biocomposites, we will be using the single walled and multi-walled nanotubes. Normally these G dash, G and D bands will be appearing for the graphite structure. You know what is a nanotube? Nanotube means it is actually folded structure of graphene. A very important peak here you can see, it is known as the RBM. RBM means ring breathing mode. So this shows that this is actually a ring. That means it is a single walled carbon nanotube. So the tube characterization you can see here, RBM in Raman, because this type of carbon nanotubes will breathe. Okay, so this breathing will show a peak in Raman spectra. So that is one of the advantages, because other type of techniques may not be useful for detecting this type of deformations and ring breathing. So ring breathing will explore that this is a tube. Okay, and also this will be very different type of response we will be getting from the multi-walled carbon nanotubes. The ring breathing will be different. Okay, suppose you have a polymer blend. How can you analyze, or is there beneficial for Raman analysis, sorry, is it some results, some important results you will be getting from Raman spectroscopy, you will see. Okay. This is actually a combined study of Raman and AFM. AFM, you may be knowing, atomic force microscopy, atomic force microscopy. I told you that here actually we are using Raman microspectrometer. That means you can also see the sample through the microscope. You can set the position. That means you can adjust the crosswire on the sample, and then you can take the spectra. So it's a very versatile technique. That means you can see the sample first. Okay, it is because of the simple sampling in Raman, because there is no separate sample holder. In most of the systems, spectroscopy systems, we cannot see the sample holder, it's inside. You have to close it. Okay, but here you can see the sample very clearly, that to a microscope you can keep your crossfire on the desired position in your sample, and you can measure the spectra from that region. So one thing is very important here. The collection area should be small. Okay, the collection area is very, very big, means you will be getting the bulk spectra. Okay, so the collection area should be very small. So that is why we are introducing different type of techniques in Raman. One of the techniques is known as the confocal Raman technique. I'll explain you. And the other technique is called surface enhanced Raman spectrometers. All these things are for reducing the sampling detection area. So sampling detection area is very small, means you are measuring from a small number of molecules. You can measure nano size. Okay, for example, a bio-nano composite, you want to concentrate on a few nanoparticles in the matrix, you can use this technique. Okay, with a picture. Okay, you see. So Raman, you will be getting a Raman image. Raman image means Raman image is resulted from the Raman signal. Here we are actually blocking the relay. Okay, so you cannot. You see, suppose you are blindly taking a spectra. When the laser light is coming, this is your sample, and simply you are detecting the spectra, means you will be getting relay scattering. Sure, because relay is the prominent scattering. You will not get a spectra because relay has no characteristic features. You will be simply getting a spectra like this. That means it's a relay scattering. So suppose you block. So you mean the important thing is that due to the light scattering, there will be relay scattering and also there will be Raman scattering. So you have to block the relay. To get the Raman signals. So you have to use a filter, appropriate filter you have to use, and when you look through the filter, you can see the Raman image. It will not be always. Suppose for you see in polymer blends, normally there will be a matrix and some the other component may be as modules you can see here. Okay, it will be distributed sometimes uniformly or non-uniformly. It's known as the heterogeneous polymer blends. The advantage is that you can measure the spectra from different regions. You can select the regions. Okay, suppose it is appearing as matrix and the dispersed system, you can measure the spectra from the dispersed system and also from the matrix. So here this PMMA PS blend, you can selectively measure PMMA and PS, and you can check the distribution of the one system in the matrix. So that is the polymer blenders. Polymer blend, what you are analyzing, the distribution of, that means the dispersion of the system, and the miscibility of the system, and what is happening in between the dispersed system and the matrix, the interface, what happens at the interface. That means some reaction is happening, some interaction is good or not. These type of things we will look into the polymer blend. So for this characterization, Raman spectroscopy will help you. See, sometimes one problem is there. Suppose you are taking a heterogeneous system, maybe sometimes it will not appear in two colors. That can be possible. Okay. In such a case, you have to stain the sample by using a Raman dye. There are special type of dye. These are common dyes, but some dyes are categorized as Raman dyes. For one example is crystal violet. Crystal violet is a Raman dye. Okay. Rhodamine is a Raman dye. So by using the different type of stains, you can differentiate the structures, so the phases. Okay. So you can analyze the filler distribution because you can select the space, select some area on the system, and you can measure the spectrum. So for example, I think it's visible or not. There are 3 points selected, point 1, point 2 and point 3, and the point 2 is the blue one, point 1 is red, 2 and 3. Actually the point 2 is showing the perfect polymer composite spectra, because in that region the polymer and the filler, all these things are coexisting. So this actually you can select, you can scan the area and to find out where the filler distribution is high and where the filler distribution is low. So this is also very easy by using Raman spectrometer, because you can take a picture also. Okay, so picture will also tell you about the regions. Okay, suppose you are going to find out the degree of curing of a resin. Suppose you are monitoring a chemical reaction, a polymerization. It's very useful because Raman will give you the time resolved spectra. That means suppose you can perform the curing, or you can perform the chemical reaction in the glass slide itself. Okay, suppose you catch the spectra 0 time, after 15 minutes or after 20 minutes, all these things you can take continuously. So continuously you can monitor. Okay, suppose you switch on the detector, switch on the software, it will continuously, it will take the spectra. That means reaction is happening on the glass slide, curing is happening on the glass slide. So you can actually, it is just like a cinema. Okay, you can monitor. Okay, the kinetics of the reaction. Okay, see what happens here. This green one is the monomer peak, and this small one you can see here is the initiator peak. Initiator is needed for the curing. Initiator and monomer peak. You can see the second one. That means after some time, what happens? The initiator peak is little bit increased, but the monomer peak, the intensity is decreasing. And after some time, that means 205 minutes of UV radiation, you can see that the monomer peak is completely diminished, disappeared. That means curing happened, or the polymerization is finished. Okay, so that means that time resolved spectra will be given by Raman. So this type of spectra will not cannot be given by other types of techniques. That means just like a film, you can watch the chemical reaction or the curing. Okay. So suppose so I told you that this is a non-destructive technique, because you have maybe in museums you have seen the ancient paintings and different type of sculptures. Uh, Suppose you want to check the material or you want to check the paint they have used, normally what you have to do, you have to scratch the picture and take some sample, or you have to scratch the sculpture and take the spectra. That is not needed for Raman, because now we have portable Raman spectrometers, and also you can focus on different areas on the sample. And suppose for a painting, for some painting is there, means you take you want to check this paint, some blue paint has been used, what is this chemical compound. You just focus the Raman beam and portable Raman spectra, and you can measure the spectra. It's a non-destructive technique, so you need not destruct the sample. So this is actually a different type of coating compound 1 and coating compound 2. So this is one coating and this is another coating. All these things you can easily find out which material has been used for this particular coating. Okay, so that may be useful for the industrial purpose, because some product is coming, every time people were asking, sir, can you find out the composition, okay, which material they have used for this purpose, all these things. Okay, but you see that we can find out that it needs a lot of chemical reaction, and also a lot of spectroscopic techniques we have to depend. Otherwise you cannot find out. Okay, suppose a product is there, a polymer composite is there, they have used some colors, some fillers, some other different type of polymers they have used. So to find out the composition is very difficult. But you see, Raman spectroscopy is making these things somewhat easy. That means it's a region wise you can measure. Suppose you have a layered type structure, it's very easy. Okay, suppose you see the difference. I think we already studied in lower classes the difference between nylon 6 and nylon 66. Okay, the nylon 6 and nylon 66 will read the infrared spectra similar. Okay, but here you see around this area you can see small peaks will be appearing and disappearing, that can actually differentiate between polyamide 66 and polyamide 6. Okay, so it's because of the conformational change and small structural change will be detected by Raman. For example, a layered plastic composites. So this can be mainly useful for the food packaging. So there are tetra layered and packaging type systems may be there. Suppose you analyze just for the IR beam, you will be getting a bulk. Spectra, maybe a combination of all these things. But here you can scan the different type of area, and you will be getting the spectra for all these things. You may be, if it is nylon, PET, all these things you can find out. You need a standard spectra, standard Raman spectra. This can be done by two techniques. One is actually you look the layer through the microscope and keep your crosswire on different layers. That is possible. That may not be possible always. The other technique is you can use a confocal Raman setup, because suppose the food packaging system is there like this. The confocal, what is the advantage? The advantage is confocal will focus, you can focus on this area, that is the top area. You can focus this area, you can focus also inside. It is not the depth depth profiling is possible. That means normally the sample should be somewhat transparent. If it's completely opaque means it's not so possible. But you see, that is the important application. Okay, I will show you the confocal setup. Okay, as our stress strain studies, also you can monitor. You see, um, uh, how we'll do this. Okay, stress strain studies, how we'll do this. You see, our sample holder, suppose you are using a rubbery material. Normally, for a tensile strength, you will be using this type of sample, sample. So suppose you can use this sample, you can measure the Raman spectra from this area, And another important thing is, you can use a holder here, and suppose you are deforming the material like this. At the same time, you can measure a spectra. So that will give information about the stress and strain. That means that will give information about the modulus. You know that it is the stress by strain at the elastic limit. Suppose for a carbon fiber, normal peak will be coming like this, around 2000, somewhere 2700. One peak only it is displayed here, and if it undergoes stress, you can see that this peak will be shifting to some other position. So this you can monitor that for every different stretching, you can monitor like this. Actually, creep you can find out like this. Okay, suppose there is a lot of points like this. So from that you can find out the modulus. Okay, so there are so many techniques like this. You can integrate a different type of setup. Suppose one sample is like this, you can just keep a weight by using a string or something. You can just keep a weight here. This weight you can change so that you can find out the creep, stress relaxation, all these things you can find out. Okay. So one of the important areas we can focus is the determination of microplastics. You know that microplastics is one of the important crisis here. Very important problem is that you cannot characterize the microplastics. That means what type of combination you are getting, all these things. But Raman spectroscopy is made it somewhat easy. That means suppose a lot of particles like this, means you can focus on different particles and take the spectra if you have a large number of particles like this. Okay, so that is also somewhat better, faster and stronger. The identification will become faster. You see, they have taken different parts of the microplastics, nylon, PET, polystyrene, PP, all these things you will be getting the spectra. And also you can analyze biofilms. This is related to the marine aqueous systems, where the some biofilms are forming on the microplastics. That means some bacterial growth and all these things, that is actually a great threat to other organisms. Okay, so that also the coating separately you can measure. That's also important. Many this type of analysis you can do. Okay, so I am coming to the confocal Raman imaging. You see how much the depth is. Okay, you see xylem and cuticle. This is the xylem part and this is the cuticle part. Okay, suppose you are analyzing a fiber, a natural fiber. So you will be getting the correct spectra from the xylem and also from the cuticle selectively. Okay, you see, suppose you have some microplastic is there. It may be a combination of different, maybe PVC, a pigment, a calcium carbonate filler, all these things. You will be getting a composite image first. This is called Raman image, Raman composite image. But from the Raman composite image, you can differentiate PVC only, that means blue only, red only, and green only. This may be mainly the color change is actually depend upon the size of the particles, size of the regions. That is also Raman will show. So you can select the regions and you can find out what are the components. Okay. See, actually confocal, what I told you that how it is working. This is your sample, this is the specimen, this is the outer surface, this is the inner part, this is the below focal plane. All these focal planes you can adjust the beam by using you can see that there are three lines are coming and three lines is actually reflecting back from the specimen from the different region. So you can focus on the top, below part, and also inside. By using this technique, you can have different layers you can analyze by using a confocal. That is why confocal is giving a better image. Okay, better image, sometimes more informative than scanning electron microscope, because here color change is also visible. So I will be one more thing also I want to tell you. I think some 10 minutes I will take, 10 more minutes I will to complete this. Okay. Raman spectroscopy is highly promising for biopolymers to analyze proteins, lipids, and all these type of biopolymers. I already told you that water is a very weak Raman scatterer, and due to that you can analyze the biopolymers in the living condition or sometimes living condition in the same environment, because suppose you dry the protein, the conformation will change, so you need moisture there. So you can take the sample as such, no need of other treatment for your sample. You can take the fiber itself, you can take the DNA sample itself, protein sample itself, you can just analyze. You can also analyze the live cell. That means cell is in a living condition. Cell may be in a living condition because you are cell you are taking in water. For maybe sometime it will be in a living condition. Suppose laser is using, the problem is that due to the laser light maybe it will die, but it will take some time. Okay. And also we actually moved to a very important another technique called surface enhanced Raman spectroscopy. That means normally Raman measurement will take a long time. That means you have to expose your sample in the laser for a long time. So that will damage the sample. That will damage your sample. So we need a very faster technique. Within seconds, we want to measure a spectra from the sample. So the very important technique is, I told you that there are so many problems are there, especially for biopolymers. One of the important problems we can face in Raman spectrometry is the fluorescence. Suppose you look through the microscope, suppose a protein you are going to analyze, you can see a very huge, very red color you can see, somewhat orange color you can see. That is actually due to the fluorescence. Fluorescence will be prominent compared to Raman signals. So what happens is, normally suppose you are measuring in conventional Raman, you are measuring the biomolecules, biopolymers means you will be only getting the fluorescence. Spectra will be hidden. So what we have to do is, you have to quench the fluorescence. That means we have to avoid the fluorescence. That means Raman signal should be very prominent compared to the fluorescence. We have to make it like that. We are actually making the Raman lines more prominent by using nanoparticles. Okay, so you may be, you know, the old, very old story. You may know, I am not telling the story. Actually, this tortoise can also fly. Okay, you know that tortoise is moving very slowly, but it can also fly with the help of birds. Okay, so Raman signals can also fly with the help of nanoparticles. So the silver or gold nanoparticles, there is a peculiarity is there. The sample, your biopolymer sample, will be absorbing on the nanoparticle, and you can measure the spectra. You can measure the spectra from the nanoparticle surface. That means your sample is not alone. Your sample is actually adhering on the nanoparticle, silver or gold nanoparticle. From that you can measure. If you measure like that, your Raman signals will be, you will be getting high intense Raman signals. This is called surface enhanced Raman spectroscopy that can be make use of biopolymers normally. Okay, you see, the enhancement is actually due to maybe it's physics, due to the peculiarity of this nanoparticles. By the way, you why the bulk gold is yellow and the nano gold is blue, green, red? Why? It is due to first one is the matter of size matter. Okay, so why when size decreases, this effect is happening? The quantum confinement is there. Quantum confinement mainly go to this when you are going to very low size, that is the quantum dot. Okay, even nanoparticle itself, this color changes, you can see. That is due to an effect called surface plasmon resonance. Because the charge will be developing on the nanoparticle. If the size decreases, that means plus and minus, in one side plus and other side minus. And suppose you are using a light or the white light, suppose you are falling on the material, there will be a standing oscillation. Standing oscillation means plus minus, minus plus, plus minus, minus plus, plus minus, like that. So there will be a standing oscillation. So this standing oscillation is making the optical effect called surface plasmon resonance. This is one of the reasons for the enhancement of Raman signals, because you are measuring from the surface of the nanoparticle. Surely you are you are actually measuring from the sample, but your sample is resting on the nanoparticle. It's not simply resting, it's absorbed on the nanoparticle. So the signals will be enhanced. So that is known as a surface plasmon resonance. By using this technique, you can analyze a living cell. That means suppose you want to study the distribution of proteins on a living cell, you want to incubate the nanoparticles on the, or you have to adhere the nanoparticles on the cell to study about the proteins. Okay, so for example, just I will show you one. I am not showing. So for that, normally one of the problems you can face here is the adsorption. That means you are using silver or gold nanoparticles. It will not adsorb on all materials. For example, protein it will adsorb, because protein consists of amide N. Nitrogen is there. Nitrogen has an affinity towards gold, so it will adsorb. For example, sulfur has an affinity towards gold, it will adsorb, but it will not adsorb on sugar. You cannot detect sugar concentration by using surface enhanced Raman spectroscopy. But you can do this by using functionalized nanoparticles, because which is your target? You you have to just check the target, and target will be binding with the functionalized nanoparticles. That means you have to functionalize the nanoparticle. You can use functionalized nanoparticles to improve the measurement. For some other, that means simply nanoparticle will not work always. Okay. So such type of a picture I will show you. So this is actually, you see, this is a yeast cell. So the protein distribution on the yeast cell is analyzed by silver nanoparticles. You can see different colors. Why different colors? See, this silver nanoparticle, but different colors. This is due to the surface plasmon resonance. That means depend upon the size, you can see the colors. Some particles may be large, some may be big aggregate. The aggregation, I think last talk you already listened. The aggregation we cannot avoid always. So the aggregate will be very high or small. So the depend upon the aggregation, the color will change. Color will change. So I will just show you the image. This is called the surface enhanced Raman image. Very beautiful image. That means this is a small, simply a white light image. But if you are using a Raman filter, you can see this type of spots. These spots is known as hot spots. So you can measure the spectra readily from the hot spot. Why? Because originally the light is coming from the nanoparticle. Plasmon effect, due to the plasmon effect, and the Raman light is directly coming from this region. So that region very easily you can measure within seconds. You need not keep your sample for a long time. That is the surface enhanced Raman technique. You see different type of, maybe this is a cell actually, but you can imagine this biopolymer or biosystem, some biocomposite system. Suppose you want to analyze the spectra from different region, you can make use of this technique. Okay, readily you will be getting the spectra. Okay, you can also perform a scanning also. A scanning. So you can select some area and you can see that this dark area and bright area. The bright area means you will be getting a spectra. Okay, so like that you can measure. Suppose polystyrene, you want to get a very good high intense Raman signal for polystyrene means what you can do is you can incubate it with a gold nanoparticles, and the absorption will be taking place, and actually you are measuring the sample on the particle. You are using laser light, there will be Raman light will be occurring, originating, and it's very easy to measure from the different regions. So I will show you the picture of polystyrene. This is the polystyrene this is the silver nanoparticle, and these are the polystyrene particles, and that will be adhere on the system, on the nanoparticle, and this is the aggregate. Aggregate means adsorbed polystyrene on silver nanoparticle. Suppose you are measuring from this area, that means on the surface of the particle you will be getting a very good spectra. Otherwise you will get a spectra, but it will take a long time and also not so intense. So you should get intense spectra within a short time. So you can improve the measurement by surface enhanced Raman spectrometer. You can use a tip enhanced Raman. That means the tip will be nano size. This, you know that this area is nano size. This is silver tip, silver or gold tip you can use, and silver or gold tip you can just touch on their sample and your laser light will be falling here, and you can measure from the tip. That means in between the tip and surface there will be sample. There will be some hot spot will be visible, and you can measure from this area. That actually decrease your area of measurement. So you can measure it from a small area, small nano sized area you can measure. You can AFM, you have used AFM. Okay, so just like this, you can scan the sample by using the tip. Then during the scanning, you will be getting the image also. Image also you will be getting, and correspondingly you will be getting the spectra from different regions. Different, very easy. That means scanning, you can give the scanning option in the software. Suppose you select an area, it will scan. You can go and take some tea and come back. Your spectra will be ready. You will be getting numerous numerous number of spectra within some minutes. You will be getting a lot of spectra. So that is one of the importance of this tip enhanced Raman spectroscopy. Okay. So you see, I told you that the scanning of the area. So this is very important for our samples, this biocomposite and polymer based systems. Suppose you want to scan some area, you will be getting the picture, and also correspondingly you will be getting the spectra. So from different area, suppose this 1, 2, 3, 4, you see that some dark area is also there. You will not be getting the spectra from that area. You will be getting the spectra from this area due to the surface enhancement, due to the nanoparticle distribution. Okay. So, but you can control this nanoparticle distribution by functionalizing the nanoparticle. Suppose very good example is you want a spectra from nucleus of the cell. So you have to prepare a functionalized nanoparticle which can attach only on the nucleus proteins. Okay. Target, you may be knowing the targeted drug delivery. Then like that, it will, when it injected to the cell, all the particle will move to the nucleus only. So if you irradiate with the laser, and if you are looking through the filter, you can only see nucleus. You can only measure the spectra from the nucleus. So like that, selectively you can do. So like that, on a composite sample or some other polymer blend or some sample, you can select the area. Some area you can select, and you can measure the spectra from there. You can scan. Otherwise, it's very, maybe you look, if it is very difficult, means you can just run a scan. The complete sample you can run a scan. Then you will be getting the spectra from different area. So that will help you to study about the distribution of fillers or distribution of fibers, distribution of additives in your polymer matrix. So this, I am just concluding the session. You see, the conclusion is Raman spectroscopy is a very powerful technique for the characterization of different polymeric systems, because the technique will give you the picture of the sample, microscopic picture of the sample, and from where you are measuring, you can select the area, and you can run a scan, and you will be getting a lot of spectra within minutes or seconds. And finally, you can, if you have all these things, results, you can analyze it very easily also, because you have standard Raman spectra for polystyrene, carbon carbonate, all these things you have. You can just compare it. You have a library of spectrums on the software. The software itself will readily tell you which component is this. Okay. So it is very easy to analyze a product, the components of a product, the pigments used, the filler used, the polymer used, all these things you can tell with less effort by using this technique. Okay, so I'm very happy that this time it's 1:30. Yeah, this time you are actually listening this talk very calmly and for your kind attention. Thank you very much. Okay.
Thank you, Professor Sujit, for such an interesting and informative session on Raman spectroscopy. We'll have a quick round of question answers session. So maybe 2-3 questions we can take.
Yes, because surface enhanced Raman platform or the substrate very easily you can make. Sputtering is one of the option. That means coating of nanoparticles. But you have to make sure that the nanoparticle will selectively adhere on the area you want. You may not be want all area. Suppose you want this area, there some chemical functional group should project. Then your nanoparticle will be adhering that portion only. You may be sputtering entire sample. Suppose you wash it, then this area will be only visible. Okay, sputtering is one of the important technique here.
Sir, first of all, very informative presentations. For me, it was useful. I have two questions. I won't say question, it's more about for my understanding. How about with the inorganic polymers? This Raman spectroscopy really works. Does it give any kind of advantages compared with other organic polymers?
Sure, but okay, question finished.
First questions. And second question is on the, you said about the portable Raman spectroscopy, where I can look at any product and see that whether this product is, what kind of components are available. So is it that being linked with like a library, means because something which we do a scanning, we need to compare with the previous items, then it will be easy for us to quickly to analyze it. So this Raman library for such kind of things are available. Even at night I was just checking with the sir also. So that will be good to know about, because I would like to use that tool for many of the competitions analysis.
Uh, it's actually came along with the software. Some spectroscopic techniques for FTIR technique, there may be a library. So the instrument itself will tell you which sample is. But I think it is advanced. Maybe I don't think that here it is there. IR library, I don't know, sir. Is it there? Or, okay. Raman, I don't know. Here it is, but it is possible because suppose already it is analyzed. All these calcium carbonate, all these things, it is analyzed. It's already there.
Okay.
Then it's very okay. That is very important. So that means it will become more effortless.
Yeah, otherwise it will save a lot of time. Because
Yeah, it will take time for the analysis. Nowadays, it will take time because you have to compare. Comparison is somewhat easy now because some softwares are there. Earlier times we just take printout and draw the scale with the scale, all these things. That is very difficult. And the inorganic particle also, inorganic polymers, inorganic particles also we can analyze, but it is coming in the low range frequency. For the CC bond, SiO2, because normally we are using the silica glass slide.
okay.
So silicon SiO bond will be reflecting, but SiO bond is at the extreme side,
Okay.
some 500 to 100 nanometer side.
Okay.
So that is why it is not reflecting. We are actually considering the fingerprint region. If you are moving to the other region, you will be getting. That is also useful, I think.
So compared with say other techniques, maybe Raman will give an advantage for such kind of inorganic polymers. Means like what you said about for advantage of Raman, like say graphene, we are seeing that we're
Yes.
able to really detect with the Raman. So same way with the inorganic.
No,
polymers also.
there are disadvantages also. but for inorganic systems. Because, but I can highlight the environmental spectra. That means in different environment you can take. You need not dry it. You need not, that type of processing is not needed.
Okay.
Okay, so environment in the same environment you can take the spectra, and also the informations vibrations you are getting less information in inorganic systems normally. Organic systems will respond more fingerprint region. The other the inorganic systems are normally out of this fingerprint region.
Thank you, sir. Thank you so much.
Sir, in the in one of the slides you showed that the silver nanoparticles were there. Above it, the polystyrene particles were coated. In tip enhanced Raman spectroscopy, the tip was over the substrate which we are going to, I mean, the sample which we are going to analyze. So, which is completely extremely different. In that case, above the nanoparticle, the sample was coated. In this case, the sample was there. Above that, the tip of the noble metal was
Yes.
scanning. So, in both the cases, how?
You will be getting high when you are comparing this surface enhanced and tip enhanced. You will be getting very good spectra in tip enhanced spectroscopy, because you are actually measuring for a very small area. The tip is very small nanometer size. This tip, so the tip actually just touches the area, or so the area will be very small. Okay, you can even go for single molecule spectroscopy. Single molecule spectroscopy does not mean that you are measuring from one molecule. You are measuring from a small number of molecules, so that the intensity will be very high. Okay, so that means it just touches the sample, and if you are touching the sample means the effect will take place. This absorption and all these things will be taking place there. At that point, we are actually introducing the laser. Actually, you are measuring from on the tip. Your sample is touching on the tip means you are actually measuring in between the sample and tip. So that will be more intense compared to the surface enhanced Raman.
So, in the tip enhanced Raman spectroscopy, the detector will be placed, I mean, at in the before cases, it is placed at the top right detector. In the tip enhanced Raman spectroscopy, will it be around? Will it be in the
No, actually, detector we are not moving. Detector is always stationary. The light, the scattered light coming from this, you are collecting. You are collecting actually, by the detector. So some light is coming from here. You have to use a filter here. Through that filter, Raman light only will come. My point is, this Raman light will be more intense. Otherwise, if you are not using tip, that light will not be intense. Light will not be intense means you will not get a very good spectra. That's all. You will be getting some noisy type spectra. So tip enhancement means you can improve the quality of the spectra. That means from a small area. Small area means a small number of molecules you are measuring.
Sir, in the spectrometer, Raman spectrometer, the laser beam is the source, or any other source, any other specific way and light we can use for different specimens like nanoparticle.
No. Laser is the light source of light here, because you know that the peculiarity of laser. This is a visible range, and also laser. When you are talking about the laser, we will say monochromatic coherent. That means the waves. You know, the style of the wave is it will be matching. All the waves are marching. In the same fashion. So laser light is, but you can use different wavelength laser. You can use a 583 nanometer laser, that will be a green laser. You can use 700 nanometer laser, that will be a red laser. And when you are moving up to higher wavelength, you can have near IR lasers. It's not visible, but it is very strong. So different range of lasers you can use depend upon the sample. The problem is that suppose you are analyzing a biopolymer, suppose you use a strong laser, means the sample will be damaging after some time. But you have to use an appropriate power laser. So before that, before you are performing the Raman measurement, you have to calculate the power density of the laser first. Then only we will decide which type of laser we will be using. I think in Raman system normally it consists of 2-3 type wavelengths. I don't know, here you can check. Maybe red, green, all these things may be there. Depend upon the sample. Suppose you are using biological sample, they will select the correct appropriate laser.
So when C.V. Raman have done the experiment, he used sunlight, no?
Yes, he first used the visible light.
So like then how he was able to?
It was very weak. It was very weak. Suppose you can also use sunlight, but you will not get a quality spectrum. The spectra should be quality spectra is needed now for the characterization. He is actually observed the light coming from the sample. He used carbon tetrachloride and other types of solvents. He first observed Raman light. That means due to the scattering, there will be intense scattering will be there. Normally consists of relay and Raman. So he used a filter, optical filter, and when you look through that filter, he will be getting Raman light. His invention is not Raman spectroscopy. Raman's invention was path-breaking invention. He introduced or he revealed Raman lines are also there. And after that only people found that the Stokes lines can be used for molecular characterization. So that is the importance of, that is why Raman got a Nobel Prize. That means it's a path breaking. Now also we are using. Okay, sunlight can generate Raman lines, but it's very weak.
Any more questions? So if there are no questions, we can wind up the session. Now I request Professor G. Unnikrishnan to hand over a token of love and appreciation to Professor A. Sujit.